everyone, ready for a deep dive? Today we're going to be tackling these tiny little defenders in our bodies, antibodies.
Antibodies,
They're
yeah.
pretty amazing, right?
Yeah.
We're going to be looking at how they work, their structure, and how they might just be a game changer for cancer treatment.
Absolutely.
Our source material today is an article from Assay Genie.
Okay.
It's called Unraveling the Complexities of Antibodies: Light Chains, Heavy Chains, Constant Regions, and Tumor-Associated Antigens.
Catchy title.
Right. So, have you ever thought about how your immune system can target such specific threats?
Well, that's where antibodies come in. They act like a specialized army, seeking and neutralizing those threats.
Yeah, and the article does a really great job of breaking it all down. For instance, antibodies are also called immunoglobulins.
Right.
They're basically these Y-shaped proteins that our body makes thanks to B cells.
Yeah, B cells are super important.
They're like the scouts of our immune system, always on patrol, looking for anything that doesn't belong.
Exactly. And those outsiders, those are called antigens. Think of them like the bad guys that our immune system is trained to fight.
Right. So when a B cell spots an antigen, it triggers the production of antibodies specifically designed to take down that threat.
Yeah, it's a really targeted response.
Okay, so it's not just like a general defense system. It's very specific. But how do they actually neutralize these threats? I mean, antibodies are tiny.
It's pretty fascinating. Each antibody is actually designed to fit onto its target antigen like a lock and key.
Wow, so it's a perfect fit.
Yeah, pretty much.
Mm.
And this binding, it can either directly block the antigen from doing any harm, or it can tag it for destruction.
Okay, so it's like a two-pronged attack: disable and destroy.
Exactly.
Awesome. Well, let's get into the structure of these Y-shaped proteins. The article mentions two main parts: the Fab region and the FC region.
Right, so think of the Fab region like the arms of the Y. That's the part that actually binds to the antigen.
Okay, so that's the business end. What about the stem of the Y, the FC region?
The FC region is like the communication center.
Okay.
After the antibody binds to its target, the FNI region interacts with other immune cells.
So it's like calling for backup.
Exactly. It triggers a cascade of events to basically wipe out the threat.
So it's not a solo mission, it's a team effort.
Right.
Okay, the article also talks about heavy chains and light chains. Can you break down what those are?
Sure. So these chains are like the building blocks of the antibody structure. Each antibody has two heavy chains and two light chains.
Got it.
And they're both essential for creating that unique antigen binding site, that lock that fits the antigen key.
And I read that within these chains, there are variable domains.
Yes.
What makes those so important?
Well, the variable domains, they're the key to the incredible diversity of antibodies.
Okay.
They're the parts that can be customized to bind to a huge range of antigens.
So it's like having a giant key ring with millions of keys, and each key can unlock a different door.
Exactly.
Wow, that's amazing. So that's how our immune system is ready for so many different threats.
Right.
But hold on, didn't the article say there was more than one type of light chain?
You're right. In humans, we have two main types, kappa and lambda light chains.
Okay, and why have two? What's the advantage?
It's all about diversity. Having two types of light chains just adds another layer of variability to those antigen binding sites.
So more options.
More options, more keys on the key ring. It just means your immune system can recognize an even wider array of threats.
So it's like having different specialists on your team, each with their unique skills and expertise?
Exactly.
That makes sense. Well, let's shift gears a bit and talk about how this all relates to cancer. The article mentions this concept called Tumor-Associated Antigens, or TAAs. What are those?
So TAAs are these unique proteins found on the surface of cancer cells.
Okay.
They're like giant neon signs that say, hey, immune system, come get me.
So they're basically giving themselves away.
Yeah, they are. And the article breaks down TAAs into three main categories. The first type is overexpressed antigens.
Oh.
These are proteins that are found in much higher quantities on cancer cells than healthy cells.
Yeah.
Like they're overdoing it,
It's showing off.
Exactly.
Okay, what about the second type?
The second type is mutated antigens. These are abnormal proteins that are created due to errors in the cancer cell's DNA. They're like unique fingerprints.
So it's like the cancer cells are accidentally giving themselves away.
Right.
What about the third type?
The third type is a little more mysterious. They're called cancer-testis antigens. These are proteins that are typically only found in male germ cells, but for some reason, they also show up on some cancer cells.
That is strange.
Just like they're trying to disguise themselves.
Sneaky. Well, it sounds like cancer cells are playing all sorts of tricks, but luckily researchers are starting to understand these TAAs, right?
Absolutely. Understanding these TAAs is actually revolutionizing cancer treatment, especially in the field of immunotherapy.
That's great.
By targeting these TAAs, we can actually use the power of the immune system to fight cancer in a much more precise way.
So it's like we're finally starting to win the war against cancer.
I'd say we're making good progress.
That's great to hear. But I am curious how we actually target these TAAs. Is it as simple as just pointing our antibodies in the right direction?
It's a bit more complex than that, but the concept is similar. Researchers are developing therapies that leverage antibodies' ability to recognize and bind to specific TAAs.
Okay, so we're taking advantage of their natural ability.
Exactly. One approach is to create something called monoclonal antibodies.
Monoclonal
These
antibodies.
are basically lab-grown antibodies, and they're designed to target a specific TAA with incredible precision.
So it's like having a guided missile system for the immune system.
That's a great way to put it.
It's pretty amazing stuff.
Yeah.
Do we have any real-world examples of these targeted therapies?
Absolutely. The article mentions a couple of examples like trastuzumab.
Shizumab.
It's also known as Herceptin, and it's a monoclonal antibody that targets the ARTNU antigen, which is overexpressed in some breast cancers.
Oh, wow. So this is actually being used to treat people right now.
Yes, it is. And there's another great example, rituximab. This one targets CD20, an antigen found on B cells, and it's used to treat certain types of lymphoma.
Okay, so we've covered a lot of ground here, from the basic structure of antibodies to their potential in fighting cancer. But before we move on, I think it would be helpful to take a step back and really delve into how those heavy and light chains work together to create a functional antibody. What do you think?
Let's do it. It's a fascinating story of teamwork at the molecular level. So yeah, picking up where we left off, those heavy and light chains, it's amazing how they come together to form a functional antibody.
Yeah, it really is like a perfectly choreographed dance.
It is. Each heavy chain pairs up with a light chain, and together they form a structure called a heterodimer.
Heterodimer. Okay, that sounds a bit complicated.
It's just a fancy way of saying two different parts working together as one unit.
Okay, so like a team.
Exactly, a team. And then you need two of these heterodimers to actually create the complete antibody structure.
So two teams working together.
Right.
And how do they know how to fit together? Like what makes sure that happens?
Well, it's not random at all. There are specific regions within both the heavy and light chains called variable domains. These play a key role in forming the antigen binding site.
Oh, okay. So those variable domains, they're what makes contact with the antigen.
That's right. And what's amazing is that these variable domains, they can be arranged in countless ways, which creates a vast library of different antigen binding sites.
So that's how our immune system can recognize and target so many different threats.
Exactly. It's this diversity that gives your immune system such incredible power.
So each antibody basically has a unique antigen binding site.
Pretty much. Think of it like this. You have millions of antibodies in your body, and each one is carrying a slightly different key. And each key is ready to unlock a specific antigen lock.
So it's not just about having a lot of antibodies, it's about having the right antibody for the job.
That's exactly right.
And those variable domains within the heavy and light chains, those are what give the antibody its incredible specificity.
Exactly. They're like custom-made tools designed to fit a very particular target.
Okay, so the variable regions handle the binding part.
Right.
But what about the constant region? We know that plays a role in directing the immune response. But how does it actually work?
Well, think of the constant region as the antibody's communication hub.
Okay.
So once the antibody has grabbed onto the antigen using its variable region, the constant region takes over.
So it's like sending out a signal.
Exactly. It's like sending out a signal flare. The constant region can interact with other parts of the immune system, like macrophages and natural killer cells.
And that triggers the attack.
Right. It triggers a whole cascade of events that leads to the destruction of the threat.
Wow, so it's really coordinating the whole attack strategy.
It's like a general directing the troops.
Okay, so one of the mechanisms it uses is something called antibody-dependent cellular cytotoxicity, or ADCC.
ADCC, yeah.
That sounds pretty intense. What happens during ADCC?
It's a way for antibodies to mark a target for destruction. So when an antibody binds to an antigen, its constant region can actually bind to receptors on certain immune cells. And this binding, it sends a signal to those cells saying, hey, this thing is bad news, get rid of it.
So it's like the antibody is putting a big eat me sign on the target.
Precisely.
And the cells that respond to this signal, like natural killer cells, they're really good at what they do.
Yeah, they release these toxic substances that basically kill the target cell, eliminating the threat.
Wow. It's amazing how efficient our immune system is. So ADCC isn't the only weapon in the constant region's arsenal, right?
Yeah.
You mentioned something about the complement system earlier.
That's right. The complement system is like the immune system shock troops. It's a network of proteins that can directly kill pathogens or tag them for destruction by other immune cells.
Okay, so how does the constant region activate the complement system?
Well, when an antibody binds to an antigen, its constant region can act like a landing pad for certain complement proteins. And this binding, it sets off a whole chain reaction.
And what's the end result of this chain reaction?
Well, the end result is the formation of what's called a membrane attack complex, or MAC.
A membrane attack complex? That doesn't sound good.
It's not good if you're the target.
Yeah.
The MAC basically punches holes in the membrane of the target cell,
Oh, wow.
causing it to basically rupture and die.
It's a direct attack.
It is. The complement system is a very efficient killing machine, and it can also enhance other immune responses.
So the constant region is really multi-talented. It not only directs its own effector functions, but it also calls in the reinforcements of the complement system.
Exactly. It's like a one-stop shop for immune defense.
Okay, so we've got the variable region, which is responsible for binding to the antigen, and we've got the constant region, which directs the immune response. But the article also mentioned that there are different types of antibodies or isotypes. What's the difference between them?
The main difference lies in their constant regions. Different isotypes have slightly different constant regions, which gives them unique properties and functions.
Okay, so it's like different types of soldiers.
Exactly, each with their own specialized skills.
Can you give me an example?
Sure. So IgG antibodies are really good at activating the complement system, while IgE antibodies are more involved in allergic reactions.
So even small variations in structure can lead to pretty big differences in function.
Yeah, it's really fascinating how our immune system can be so adaptable.
Okay, so we've got these different antibody isotypes, each with its own special abilities. And we know they play a crucial role in fighting off infections, but I'm curious about something else the article mentioned. It said that antibodies can cross the placenta. Why is that important?
Well, that's a really interesting aspect of antibody biology and has to do with protecting newborns.
Oh, okay.
You see, babies are born with a relatively immature immune system. They haven't really had a chance to build up their own defenses yet.
So they're pretty vulnerable.
Exactly. And that's where maternal antibodies come in. During pregnancy, certain antibodies, like IgG antibodies, can actually cross the placenta from the mother's bloodstream into the fetal circulation.
So the baby gets a dose of the mother's immunity.
That's right. It's called passive immunity, and it provides the baby with a temporary shield against pathogens that the mother has already encountered.
Wow, so it's like a head start for the baby's immune system.
Exactly. And this protection lasts for the first few months of life, which gives the baby's immune system time to mature and develop its own defenses.
That's really amazing. It's like a gift from the mother to the child.
It is. It really highlights the critical role that antibodies play, not only in fighting off infections, but also in ensuring the survival of future generations.
Okay, so we've covered a lot of ground here. We've explored the structure of antibodies, their different parts, their various functions, and even their role in protecting newborns. But I want to bring it back to cancer for a minute and talk about how all this knowledge is leading to new and innovative treatments.
That's a great point. Remember those tumor-associated antigens, or TAAs, we talked about earlier?
Those flags on cancer cells?
Right, those flags. Scientists are using that knowledge to develop targeted therapies that leverage the power of antibodies to fight cancer.
It's like we're finally giving the immune system a weapon it can use against cancer.
That's a good way to put it. One approach is to create something called monoclonal antibodies. These are lab-grown antibodies that are designed to target a particular TAA.
So it's like a custom-designed weapon tailored to attack that specific type of cancer cell.
Exactly. And these monoclonal antibodies can actually work in a couple of different ways. They can directly kill cancer cells by triggering ADCC, or the complement system, or they can actually block the growth and spread of cancer cells by interfering with their signaling pathways.
Wow, so there are a couple of different ways they can attack.
Yeah.
It's amazing how precise these therapies can be. But I'm wondering, are there any challenges in developing these targeted therapies?
Of course, there are always challenges in science. One major challenge is actually identifying TAAs that are truly unique to cancer cells. We need to make sure that we're not targeting antigens that are also found on healthy cells.
Right, because that could lead to some pretty serious side effects.
Exactly. Another challenge is actually making sure that the immune system is effectively activated to actually destroy the cancer cells.
Yeah.
Sometimes cancer cells can be really sneaky, and they can evade the immune system.
So it's not just about delivering the antibody, it's about making sure it can actually do its job.
Exactly, and that's where a lot of the current research is focused.
Well, it's incredible to think about the complexity of the immune system and how scientists are constantly unraveling its secrets to develop new treatments for diseases like cancer. It gives me a lot of hope for the future.
Me too. It's a fascinating field, and the discoveries that are being made are truly groundbreaking.
You know, it is amazing to think about how much we are learning about the immune system
It really is.
and how it can be harnessed to actually fight disease.
It's a truly remarkable system,
Mm-hmm.
and the more we uncover, the more possibilities kind of emerge.
Yeah, you know, at the beginning of this deep dive, I have to admit, I was a little intimidated by all the technical terms, heavy
Yeah.
chains, light chains, variable domains.
Yeah.
But now it all makes so much more sense.
It can be a lot to take in,
Right.
especially when you consider how complex the immune system really is.
Right.
But I think that's the beauty of these deep dives.
Yeah.
We get to break down these complex topics into bite-sized pieces and really explore what makes them fascinating.
Absolutely. And I think this one has really driven home just how important antibodies are,
Absolutely.
not just for fighting off everyday infections, but also for treating some of the most challenging diseases we face.
Yeah, the potential for immunotherapy is huge.
Right.
And we're only just beginning to scratch the surface.
Yeah.
You know, I'm particularly excited about the research being done on using antibodies to treat autoimmune diseases.
Oh, that's interesting. Can you tell me a little more about that?
Sure. So in autoimmune diseases, the immune system mistakenly attacks the body's own tissues.
Right.
It's like friendly fire and can cause a lot of damage.
Right. I know conditions like rheumatoid arthritis and lupus are autoimmune diseases.
Exactly. And researchers are exploring ways to use antibodies to kind of re-educate the immune system
Okay.
and stop it from attacking healthy cells.
So using antibodies to restore peace.
Exactly.
That's incredible.
Yeah, and there's also a lot of exciting research being done on using antibodies to deliver drugs directly to target cells.
Okay.
This could be a game changer for treating cancer because it would allow us to deliver high doses of chemotherapy directly to tumor cells while sparing healthy tissues.
So it's like a guided missile system for drug delivery.
Exactly.
Wow. Sounds like the possibilities are pretty much endless.
They really are, and I think that's what's so inspiring about this field.
Yeah.
There's so much potential for discovery and innovation, and it feels like we're on the cusp of some major breakthroughs.
Well, I, for one, am excited to see what the future holds for antibody research, and I hope this deep dive has sparked some curiosity in our listener as well.
Me too.
Yeah.
It's always rewarding to be able to share this kind of knowledge about such a fascinating field.
Yeah, this article from Assay Genie has been an excellent guide on our journey into the world of antibodies.
It really has.
We've covered so much, you know, from the basic structure to their function to even their therapeutic applications.
It's been a pleasure exploring these concepts with you, and I really encourage our listeners to continue learning and exploring. There's a whole world of knowledge out there.
Well said. Thanks for joining me on this deep dive into antibodies. Until next time, stay curious.